All right, so we're going to start with the first talk, which is Jan Lötvall. So he actually doesn't need introduction, but I made him to write a short bio so I can read it. So Jan is a professor at the University of Gothenburg, Sweden, and he has extensive experience in the field of translational asthma research. So for the EV field, he is famous for showing that exosomes carry and shuttle functional RNA between cells. Jan was the first president of ISEF between 2011 and 2016. And for the past half a year, he is the editor-in-chief of Jeff.
Thank you, Carolina. I'm presenting a little bit about extracellular vesicles and their subpopulations, and specifically on some work on mitochondrial proteins that we published last year in JEV. I just wanted to start by showing a picture that Rossella Crescitelli in our lab has taken. This is a bronchial biopsy from a healthy young human person. And you can see a cell here. This is chemically fixed tissues. You can see collagen fibers, and you can see a lot of small round structures in the interstitial space, and they can be quite large in some instances or smaller. So if we look with a little bit greater magnification, you can see here vesicles of different sizes, and here's one that actually looks like a whole mitochondrion, and this is not the first time that has been illustrated, but you can also see smaller vesicles, maybe all the way down to the 50 nanometer diameter size. So this is just illustrating that, firstly, you can see in tissues a lot of vesicles, but also they have a lot of different morphology. So thinking about different types of vesicles, different subpopulation of vesicles is very important when you're doing EV research. So thank you for introducing me, Carolina. As you mentioned, I'm professor of allergy in Gothenburg in Sweden. This is declaration of interest. I have multiple patents in the field of extracellular vesicles, and some of them are at Kodiak Biosciences, where I actually worked for almost 2 years as chief scientist. I'm now back in Gothenburg. I've started a small biotech company with some seed funding called Exosomes Biosciences, and I do some consulting through another company called Vesicle Biosciences. And as you mentioned, I'm editor-in-chief for Journal for Extracellular Vesicles. When this is very exciting, of course, JEV has an impact factor of 11 right now when we're getting a massive amount of submissions to the journal, so we have to be quite tough in our selection process of publications. So I'm going to present a lot of work primarily from our own lab, not so much an overview, and I think that's the intention of this series, not a general overview, but actually speaking about your own work. And Suchul is a very important person for, there we go, laser pointer, for this work. So he's presenting some of his data, but also Rossella Crescitelli has been working a lot on isolating vesicles, for example, from tissues, and some of that work will be presented. Taral and Alexander have been involved, and of course Cecilia, whom many of you of course know. I want to emphasize that our lab was very much inspired over the last 8 years or so by Yong-Song Go, who's a professor at Postech, the university in South Korea in Pohang. And he's a really brilliant mind and has been thinking about extracellular vesicles for longer, probably than any of us at this meeting. So Yong-Song has been a great inspiration. He was also a guest professor in my university for a period of time. So when we think about extracellular vesicles in general, I'm trying to get that off the screen. We have classically been talking about microvesicles that are considered to be released from cells through budding of the membrane. And it's believed that those are isolated through the little bit slower certification pace of 10,000, maybe 16,000 G in different types of protocols. And then we have these vesicles that come out from the inner part of the cell, from the multivesicular endosomes, and are released when these endosomes, multivesicular bodies, are fused with the cell membrane. And these are considered to be smaller vesicles, and they're isolated with 100,000 Gs. And they're typically called exosomes when they're prepared. So I should say that no current separation method separates vesicles that are released through budding or vesicles that are released from the multivesicular endosomes. So there are it's probably impossible to totally separate them from each other with the current technologies, but we do see different profiles of vesicles that are isolated through the 10,000 G centrifugation or the 100,000 G centrifugation. And the interest we have in vesicles is that they can do things. They can mediate biological signals. They are much more complicated than hormones or nerve signals, for example, or cytokines, because they carry a lot of different molecules. They have a lot of molecules on their surface, and they have a lot of molecules on the inside. And our contribution, as you mentioned, Carolina, was that they carry functional RNA and can shuttle that RNA between cells. So when you start thinking about what vesicles in general, all these vesicles that are released by a cell can do, you realize that they can do surface signaling. And I'm not going to talk about that so much today, but that's probably an important component that we can measure easily. So you can see phosphorylation signals happening within maybe 5 or 10 minutes after putting vesicles into cell culture. But they can also deliver proteins to the cytosol of the cell or deliver RNA. And perhaps some of those mechanisms, some of those functions of the vesicles are a little bit slower and perhaps even more difficult to measure with standard scientific tools. So I think there's a lot of work to be done to really understand the full extent by which vesicles mediate biological function. So should move this a little bit. There we go. So when you work with extracellular vesicles over multiple years and you start isolating vesicles through different types of centrifugation, you realize that there are differences in the vesicles, and the concept of each cell sending out an array of different types of vesicles becomes kind of natural to think about. And so in principle, the concept that we are building the science on is that both the microvesicles or the larger vesicles and the smaller vesicles probably contain a lot of subpopulation of extracellular vesicles. A lot of the work we're currently doing is focusing on that type of research. So then you realize if a cell is sending out these vesicles, and when we isolate these vesicles, we put them on another cell culture, for example, or into an animal or whatever we're doing, we get a signal or we get an effect or a phenotype change. But it's not so easy to understand exactly what that phenotype change depends on. So of course, these different subpopulations can mediate multiple biological functions, and all of these different possibilities could contribute to the biology that we see. So as you know, explaining a biological function through a single molecule is perhaps not always that easy. So the work is definitely looking at different subpopulations right now. Cecilia and Nasibi in our lab are pushing for that in human blood, and there will be a lot of interesting information coming out from that later this year, hopefully. So 2 years ago, Cecilia, Suchul, and I published an overview paper discussing the different ways by which we can think about subpopulations of extracellular vesicles, and this was published in Molecular Aspects of Medicine that year. So, and we also discussed the possibilities of different vesicles actually having different therapeutic functions. So in an ideal world, maybe you should isolate a specific subpopulation of vesicles to have a specific therapeutic effect. So we divided this into biogenesis, density sedimentation, different RNA cargo, different protein cargo, size, and morphology. And I will start by discussing morphology. And I think there was a eureka moment when we started looking at extracellular vesicle with cryo-electron microscopy, which is a method by which we can visualize the vesicles without doing any chemical stain or anything like that. This is just one example of work from Johanna Hög, who was in our lab for a few years and helped us really visualize the vesicles in different ways. So she's an amazing electron microscopist and loves cryo-electron microscopy, and started characterizing and dividing the vesicles in different categories. And she did that in this case with Davide Sabeo, who is her PhD student. So a majority of vesicles are what we conceive as extracellular vesicles, a standard extracellular vesicle, which is a single membrane, single vesicle. But there are many other vesicles there, and this is looking from a single cell type. This is from HMC1 cell, which is a mast cell line that we worked a lot on. So there are double vesicles of different types with different sort of membrane separations, and sometimes the electron density you see here, that the inner vesicle inside of the larger vesicle is actually more electron dense, and you can see different patterns in that perspective. You can also see small double vesicles, and you can see oval vesicles, tubules of different sizes, incomplete vesicles, so those are sort of open vesicles, and long tubules. We also see these quite extensive, it's very difficult to visualize maybe in this EM, but very long vesicles with actin, what seems to be actin filaments inside. And when we, let's see if we can show this video. No, I couldn't show this video, but we can see vesicles that actually have some ability to change, to move their membrane around. And that's probably vesicles that have these actin filaments inside. So Johanna and her student divided these vesicles into different categories. And when you really dig deeply into the morphology, you can maybe identify up to 80 different types of extracellular vesicles. So morphologically, there are a huge diversity of extracellular vesicles, and are they containing different molecules? Are they mediating different functions? That we may not be able to answer yet. So when we look at density, sedimentation, and size, we know that higher spinning, the 100,000 G isolates more of the smaller vesicles, and the less intense spinning isolates microvesicles or perhaps larger vesicles, regardless how they actually were produced by the cell. So, and we've shown and seen that these type of isolations actually isolate vesicles with different nucleotide cargo. And I'm just giving one example of that, and this is our work from 2007, where we used the 100,000 G pellet to look into the RNA cargo, and we could see primarily small RNA and no ribosomal RNA peaks in the exosome isolate. And when we did Affymetrix analysis of those vesicles, this is mRNA data, we could see that the cellular mRNA and the exosomal mRNA was not correlating at all with the cell. So there seems to be a specific packaging of exosomal RNA into the exosomes. This is in sharp contrast with what you see with these vesicles that are isolated by a slower spin. so the microvesicles. And this is work from Yong-Sung Go that was published well before I knew him personally, back in 2009. And here you see that the cellular RNA profiles and the microvesicular RNA profiles are quite similar, with mitochondrial RNA peaks in the microvesicles. And when they did a analysis of the specific RNAs inside, mRNAs inside the microvesicles or the cells, there was a very close correlation. And when you look at different articles, you may find people that claim that exosomes and cells correlate, but when you look at their isolation method, they've almost always used a type of microvesicle isolation procedure. So this has been further analyzed by other people in the lab. This is work that we collaborated with Andrew Hill and Leslie Cheng in Melbourne, and Taral is first author on this paper, where we looked at different RNAs, in this case, small RNAs, microRNAs, in the exosome isolates, the microvesicle isolates, the apoptotic body isolates, as well as the cell. And as you can see, all of these different centrifugation methods isolate structures with somehow different RNAs. So again, suggesting that there are different subpopulation of vesicles actually carrying different RNA. And the principal component analysis in this case shows clearly that these three isolates are distinct and separate in this analysis. So I'm not going to go through so much into biogenesis, but I think understanding biogenesis in detail of extracellular vesicles, we have to understand their subpopulation. There's some excellent work from Clotilde Théry that were published years ago that show that Rab27A and B can be involved in exosome biogenesis. But when you look carefully into that data, you don't eliminate all of the extracellular vesicles by knocking out or knocking down these molecules, but you see about maybe 50% reduction. So understanding the regulation of production of different populations from different cells and by the different molecular mechanisms, you would need to understand and be able to quantify, I guess, the subpopulations in an efficient way. So our next approach to understand subpopulations has been to dig into the protein cargo, and specifically regarding surface molecule, with the bias or the thought that extracellular vesicles are subpopulation of extracellular vesicles are clearly distinguishable by their protein cargo. That might not be 100% true. There could be other components such as RNA or morphology, for that matter, that distinguish different subpopulations. But in our current work, this is the path we're taking to understand that work. So the concept is that even though these vesicles kind of look the same, they may have totally different molecules on their surface, which of course may reflect in different cargo molecules as well. Because when you do detailed analysis of the content of extracellular vesicles, you can find thousands of different proteins and many hundreds of different membrane proteins or membrane-associated proteins. So there's clearly a huge diversity of extracellular vesicles. So we pursued some work with a specific curiosity, and that was whether there are a subpopulation of extracellular vesicles that carry mitochondrial proteins, because we kept on seeing a little bit of these mitochondrial membrane proteins in our different analysis that we did. So in this case, this is EV membranes from the mast cell line that I mentioned before. And when you look at the percentage of distribution of different membrane proteins, a majority is plasma proteins. Actually, a significant amount of proteins are from the endoplasmatic reticulum, and you can even find some Golgi proteins. In this case, we didn't see so many Golgi proteins. Otherwise, other ones can, in other analysis, you can see perhaps more. But we clearly saw a cluster of proteins that are associated with the mitochondrial membranes. And it was not any mitochondrial membrane. It was actually mitochondrial inner membrane, not the outer membrane. You could find a few outer membrane proteins, but it's mostly the mitochondrial inner membrane proteins that we could observe in that case. So Suchol pursued this work energetically and professionally. He's well-trained. He came from Yongsong Go's lab in Korea. So he separated vesicles to different means and specifically to determine whether subpopulations existed. And he made a bead that carried the molecule called MTCO2, which is a mitochondrial inner membrane protein. And then he enriched those, and we did proteomics analysis, membrane proteomics analysis of these vesicles that were captured on these beads. And you can see a much deeper and analysis and deeper information about proteins in those vesicles. And when we looked at those proteins, many of them were definitely associated with the mitochondria. So important as well is that we looked at ATP synthase activity in the vesicles as a whole or in these vesicles. It's been known that vesicles can carry ATP synthase activity for many years, probably decades. I think there's some work even in the 70s that imply that. And but in these vesicles, with the MTCO2 protein on the surface, we could see increased such activity. So this argues that a subpopulation is enriched in mitochondrial proteins, but also carries some of those functionalities that mitochondria have. So as I said, Suchol worked hard to try to dissect the subpopulations of these vesicles. And he performed a whole series of different direct ELISA analysis. So what he did here was to put beads into the wells and to the plastic, and then basically develop an ELISA that for different components of membranes from different cell components of membrane proteins, and then look at luminescence. And in this case, we have multiple types of samples. So we have, in this case, 3 melanoma tumor tissue vesicles. So this is tumor tissue vesicles based on methodologies that Yongsong transferred to our lab, and Rosella Crescitelli perfected and published earlier this year on. So we can see here that in these human tumor tissue vesicles, there are 2 cases here that have a lot of COX-6C, but also MTCO2. So you can see these 2 patients that there's MTCO2 and COX-6C. So MTCO2 is produced by the mitochondrial genome and COX-6C by the nuclear genome. So is clearly complementary from that perspective. We also looked at some other proteins. In this case, we could not find this protein in cell culture. This is a melanoma cell line. This is a mast cell line, and this is mesenchymal stem cells. And we could not see this protein in those instances. Whereas we could see a little bit of COX-6C and MTCO2 in the HMC1 cell line. And then we look, so these are the mitochondrial proteins, and here we have cell membrane. They are also present in the tumor tissue derived vesicles, endoplasmatic reticulum, and again, endoplasmatic reticulum. We never really pursued the ER subpopulation of vesicles in these tissues, but I think there's definitely an opportunity of pushing for that understanding further. What we did based on this data was to develop a sandwich ELISA system to specifically look for MtCO2 and COX-6C co-positive subpopulation of vesicles. And again, to analyze that, we used a sandwich ELISA. We did not yet, at that time, have single vesicle analysis capabilities. So basically, we used an ELISA. And you can see here from these two tumor tissue-derived vesicles that there were vesicles that were positive for both MtCO2 and COX-6C. So we could set a signal in these tumor tissues, whereas they were not so clearly present in the different cell culture-derived vesicles. Now, if you look at MtCO2 and CD9, you could see a lot of those in the mast cell line vesicles, and even some in the mesenchymal stem cell derived EVs, whereas this combination was not measurable or clearly very low in these other samples. So, and then we did other combinations, and again, it was difficult to find or identify those subpopulations. So we continued with that, and we pursued a larger number of patients where we could see that in the tumor tissues, there were clearly measurable MtCO2 and COX-6C co-positive extracellular vesicles in those tissues. You could measure them in HMC1, a little bit in mesenchymal stem cell vesicles, but HEC293 cells did not seem to release vesicles with this combination of proteins. So that is distinct, suggesting that different cells actually release and produce different vesicles with different subpopulation of extracellular vesicles. So we pursue this work and started looking into tissues and into plasma, and specifically we compared in plasma the relative luminescence of these COX-6C MtCO2 positive vesicles compared to melanoma, and there are much more individuals in this group, but we did get a significant difference between these two groups so that suggests this is not a diagnostic tool. It is, if anything, a diagnostic candidate, but it's interesting and suggesting that subpopulation of vesicles potentially could be diagnostics in different diseases, in this case, metastatic melanoma. So there are, of course, other opportunities looking at different types of proteins, tumor tissue-specific proteins, different RNAs, different DNA as well, and of course, mutations. So that's clearly another many alternatives or opportunities in developing diagnostics for different diseases. So this is a conclusion slide. Clearly, vesicle cells are releasing multiple types of extracellular vesicles into the cell culture medium or into the tissues. They carry a lot of different membrane proteins and also different RNA cargos. So this example of mitochondrial inner membrane vesicles is just one example of a subpopulation. I think there are many more subpopulations that we can discover in the future. So of course, unfortunately, the ICEL meeting in 2 months is postponed, but there will be a web meeting. So there will be things happening at in probably Philadelphia during that time, and through the web there will be presentations and discussions. So please consider joining that work as well. And of course, please consider Journal of Extracellular Vesicles for your best EV research. The impact factor is good, it's not going to go down. So think about Jev when you're submitting your work. Thank you very much for your attention, everyone.
Thank you so much, Jan. It's very interesting talk. I have one I have one question that I really want to ask with this mitochondrial inner membrane. So does that mean that it's not bilayer?
So presumably that means it's not bilayer, because it's not necessarily that the double membrane vesicles are mitochondrial. That's not a conclusion we can draw from this. And we saw very few, if any, mitochondrial outer membrane proteins in these vesicles. So it seems that it's specifically the inner membrane that somehow perhaps interacts with multivesicular endosomes to produce this subpopulation.
Yes, and so nearly like even close to 30% is mitochondrial
30%
membrane
of the proteins,
yes.
Right? Yes.
So,
and that may be different in different tissues, different cells, and so on and so forth. So whatever is your favorite model to study, you have to do the science experiments yourself to figure out which subpopulations are present in that situation.
Okay. Is anyone want to ask questions? Okay, Sheriff Ibrahim, would you like to speak up and question Jan directly?
My question is about, first, thank you so much for this nice and informative talk. My question is about the morphology of the EVs under the cryo-electron microscope. And if there's a possibility that the method of isolation could affect the morphology, as the manipulation could cause change and how these vesicles look like.
Yeah, so that's a reasonable question to ask, and yes, it's possible that the method of isolation could explain some of the morphology, but actually a paper before that paper that I just mentioned, and Johan published a paper where she visualized vesicles in human ejaculates, so basically prostasomes or what you would like to call them. And there was absolutely no isolation method. So that biofluid is extremely rich in extracellular vesicles. So she just took that fluid, put it in the cryo-electron microscope, and could see vesicles. So she did that not for the purpose of looking at vesicles, but actually for understanding the sperm tail in more detail. But there was no isolation method whatsoever. And when she did that, you can see exactly the same type of vesicles that you see in the isolation. So I think the isolation method, yes, it could. But, so primarily, if you want to be very careful when you're doing this type of experiment, perhaps you should be using a swinging bucket rotor with a cushion on the bottom, a density cushion, so you don't have that impactation of the vesicles toward the plastic wall of the tube. You don't have them cramped into a pellet. You actually have them floating in liquid, and that might be a better way of isolating them, specifically when you look at morphology.
Okay, thank you so much. Thank
you. We have a next question from Pia. Hi, Pia.
Pia needs to unmute.
Pia needs to do a lot of things. Hello. Yeah, so what do you think that why are melanoma cells shedding these type of vesicles that have the mitochondrial inner membrane and outer membrane proteins? So what would be the function for these?
So yeah, that's of course interesting. I should say from the beginning that it's not only melanoma cells that do it. Actually, we saw more of these in tumor tissues than we saw from a melanoma cell line. So it's not a specific melanoma subpopulation. Every single one of us on this call have these subpopulation of vesicles circulating in our blood. So, we could measure them at baseline in healthy individuals. And that does not mean that we have have melanoma. So probably multiple different types of cells do it. How are they produced? I actually don't know. We have captured an EM picture, again from a healthy individual, where we saw a mitochondrion being very closely associated with a multivesicular endosome. It seems that they were sharing membranes, and specifically the inner membrane of the mitochondria seemed to be associated with a multivesicular endosome. And somebody told me that that's called mitophagy, perhaps, and that might be a process by which these are produced. Do you have any other suggestion?
No, I'm just thinking more of the function. That is it something that if we think that well, now that you told what you told, then this might not apply. But I was thinking that the cancer cells want to survive. So, and usually apoptosis is involved with the mitochondrial. So maybe somehow the breaking down mitochondria, which are releasing the apoptosis initiating proteins, they should be removed. So it would be a sort of like a protection or tidying up process or something like that.
That makes sense. I should emphasize that the mast cell line we're working with has an extremely high viability. It's almost impossible or very, very difficult to find a dead cell in those cell cultures. It's well over 99% viability in most instances. So we don't believe that apoptosis, ongoing apoptosis in the cell cultures is responsible solely for that subpopulation. Now in health, in our blood, we have those vesicles. Of course, we have apoptosis going on in our body all the time, and it could very well be a contributing factor for those vesicles being present in the circulation.
Yeah, thanks, Jan. It was a really great talk.
Thank you.
Thanks, Pia. And then Bas, would you like to follow that up?
Did you look at RNA in these vesicles?
No, we did not look at RNA yet, as I know. So that's definitely, there's so much more to be done, right? So
it would be very
interesting.
I think it would be very interesting to look if you are planning to look at RNAs. To look at the length of the products that you find in there, because what we've seen,
the length of the RNA fragments.
Well, if they are intact, if it's whether it's debris or so random fragments that you see there, or whether it's really transcripts or processed RNAs, you can see that if you analyze the fragment length. So it could reflect clearing of degradation material through.
So
I know you don't like the garbage theory, but we see.
No, there's a garbage is. I mean, how much garbage do we throw out of our houses every day? A lot, right?
Yeah.
And but so does our body.
There's a lot of information. I know exactly.
Yeah, and it doesn't necessarily mean that the garbage process is not absolutely crucial for life, right? We need to get rid of things. Cells need to get rid of things, and it's very possible that the mitochondrial inner membrane subpopulation is part of such a process. I'm not questioning that. The one thing the comment I would have on your question, which is we do see mitochondrial RNA in mitochondrial DNA, I should say, in vesicles, and we don't know whether that DNA is actually present in the subpopulation of vesicles. And I think that's something we should think about doing. I see Rosella is here. Maybe we should isolate vesicles to do that, Rosella.
Yeah, I mean, also the fact that you find your membrane proteins intact on the surface of the vesicles is also a really interesting finding. I mean, how does it end up there? It's very puzzling, at least to me. But cool. Well, food for thought, Jan. Good luck.
Food for thought.
Thanks.
Yeah. Thanks.
Okay. We have next question from Kalyani. Kalyani.
Yes, it was a great talk. So I wanted to ask you that I think when you have isolated the subpopulation, and one of the subpopulation is the incomplete vesicles. So have you studied further about these incomplete vesicles? And I want to know that they solely express the surface molecules, or I mean, if they are incomplete, if their membrane is incomplete, so how would they encapsulate the RNA or the protein cargo?
So the incomplete vesicles I spoke about was with the cryo-electron microscopy pictures, and we can see those type of vesicles in the cell supernatant or in the body fluids. I don't have any information which surface molecules those incomplete would have, if they're different than others, or if they just damaged vesicles. It's quite possible. So I really can't answer that interesting question, but it's tough to. And of course you can do electron microscopy with antibodies with cryo-electron microscopy, but it's not done so easily. It's very extremely time consuming to do. But potentially you could have antibodies with different size gold on them to try to identify which molecules are in different vesicles, and that's definitely an interesting question to pursue, but it's tough work. And you need a cryo-electron microscope, and we don't have one. So.
Okay, thank you. You're really doing an impressive and a very intriguing work. Thank you so much.
The first question, I have two questions actually. The first is, in your experience, you have been working on this EV heterogeneity since a long time. So I think the role of the idea that EVs contain also mitochondria, or that some of EVs might be naked mitochondria, is coming up. So what do you think in your experience? And then the second is just a general question, comment. So what's next step in understanding EV heterogeneity? So what do you think?
Yes, I'll comment on that. So so there are clearly a lot of work appearing with cell-free mitochondria being released in different way, being present in different tissues and circulation. I showed you a picture of a whole mitochondrion as well. The difference there is, of course, they carry the outer membrane proteins, and in our subpopulation of vesicles that we enrich, we do not see much of that. So there are probably multiple subpopulations of mitochondrial enriched vesicles as well. So could be whole mitochondria and inner membrane enriched mitochondrial inner membrane enriched subpopulations. So there's definitely more work to be done that. The last question you asked was, how do we pursue this work towards the future? How do we really understand these subpopulations in a better way? So I think there are two things we need to do. And first is we have to sort of build data to have a reasonable hypothesis what subpopulations exist. So I throw out there's an ER subpopulation of vesicles, probably. We haven't pursued that yet, but we could. But our data suggests that there could be very well be an ER vesicle as part of the vesicular secretome cells. Now, once you document that, probably through beads and membrane proteomics or proteomics analyses of these vesicles, then you can develop assays to look at them. What we did a few years ago now was to do sandwich ELISA. But the current state of the art, or the current maybe beyond the state of the art, is to look at single vesicle analysis, right? Single vesicle analysis as a thing by itself is not interesting. It's really not interesting. But if you know what you're looking for, if you're looking for a subpopulation of vesicles with two different proteins on their surface, and you can quantify that, and you can get information on how big that percentage is, in your vesicular isolate that you're working with, then you learn something. So, okay, the mitochondrial, I'm just guessing, I'm just throwing out of it. Maybe they're 5% of the vesicles from a certain cell type, and there's 0 from another cell type. But then you can start looking at mechanisms of production. You can start knocking down things, and you can really count on those things. So that's where I think single vesicle analysis can help us to understand subpopulations in a much clearer way. So it's a little bit like the development of the immunology field. So when I started in immunology, there were T cells, and then there were CD4 and CD8 cells, and T helper cells of different types, and T killer cells, and what have you. And now there's subpopulations of T cells that are emerging very rapidly. But here we probably have a tool by which we can identify our subpopulations by surface molecules, and by doing that we can take the EV field sort of forward in a similar way that immunology has evolved over the last few decades in a positive way. So I think that's an important opportunity.
Yeah, so to have like a fact sorting or beads or things like this would be really helpful.
Beads is definitely important for analytic purposes, and then you have single vesicle analysis, and you can do the ExoView, or you could do the nano FCM. There are probably other tools out there as well that could be used, and we're pursuing that work now. So as soon as we get back to the lab full speed, we're going to look more into that.
Sure. Okay. Thanks.
Okay. Thanks, everyone. I think we need to keep it about an hour or so. So I just have a couple of slides. So I'll thank you very much, first of all, for Jan. That's interesting.
Before we end, I would just want to say that this lecture, we'll put up on a YouTube channel. I am also going to have some sort of podcasts where I speak with people and don't go into data, but actually talk about their thinking about extracellular vesicles and how they got into extracellular vesicles and things like that. So I did one with Carolina. We had a little bit of a failure when we recorded it, and we might do it again. But I also have other people, and that will be something appearing in the channel as well. So please feel free to listen to those if you're curious.
Okay. Just a couple of information. So what I share right now is our next session, which is like will be done on Monday, Tuesday. So this is more suitable for US, but also suitable for Asia Pacific. So Suresh Pativanan, very well known in the EV field, he's going to give a talk and will be hosted by myself and Dolores Divisio. And just to make sure that everybody understand that for 2 different regions, we have different meeting ID. So I'll make that clearer in Twitter and Facebook posts. And again, if you like to present, please email. So I created the email address for this, and or you can direct message me on Twitter. Remember to say your name, location, title of your talk, and also help us spread the word. So you can use the hashtag WebEVTalk. All right. Thank you so much, everyone. That's wrap up the whole session. Been a great one. It's our first trial, so hopefully we get our recording pad done right. And
yes, and then it will take a few days, but hopefully we'll have it on YouTube, and then we'll post that on the Facebook group and through Twitter, and ask for your help to distribute it further to the scientific community.
Yeah. So I might
thanks, Carolina, for taking this initiative. It's fantastic.
That's cool. Yeah, so maybe I'll see some of you on Tuesday, but see you again on Thursday for the Europe session, Europe Asia Pacific session. Okay, bye-bye, everyone.
Bye-bye.